So yeah, I'm gonna start giving you a general introduction to ontologies and specifically the gene ontology, and then my colleagues will explain annotations and other aspects, and ending with enrichment. So yeah, using the gene ontology generally and also the annotations in UniProt. This is the focus of this course. So the objectives are to become familiar with the gene ontology. Maybe some of you already are. Distinguish the different types of Go annotations. So the ones you probably are familiar with are the standard Go annotation, but for several years now, we have worked on this Go-Causal Activity Models, which we will describe in detail. But now we're starting to have like a pretty decent corpus, and it's starting to be interesting to do various types of data analysis. In addition to the standard, Go annotation analysis that people are familiar with. We'll also explain how you can find Go data in three main resources, QuickGO, AmiGO, and UniProt, and how we do phylogenetic annotations in order to gain like broader insight on gene function across different phylogenetic range of organisms. And finally, we're going to go over the enrichment analysis technique, which is really a technique that many people are familiar with for Go ontology. And it's kind of the killer app. So we want to just give some introduction. It's not going to be in depth. I know this offers enrichment analysis courses. So this is not the focus of this course, but just to give you a glimpse of what you can do. Okay, so the first part. What is an ontology? So an ontology is, really just a set of concepts and category within a subject area. So it includes the description of those concepts as well as relationships between those, objects or concepts. And so it's essentially a logic-based way to organize knowledge. So this is a bit different from there's other SIB courses describing like controlled vocabularies and trees and other type of knowledge organization, but the ontology is really a logic-based structure of knowledge. So why are ontologies useful? So this, like ontologies, I also want to say this is way beyond biology, but today, of course, we're focusing on gene ontology and a little bit outside that, but generally, even beyond biology, ontologies are useful, first of all, to resolve ambiguities. Like sometimes, depending on your expertise, the same concept like the same word or the same name may represent a different concept to you. So we need to be able to describe these things precisely. So one concept can have many ways to describe it. So for example, this is gluconeogenesis, the synthesis of glucose, and you can find it in textbooks or in various papers under these different names. And so for, somebody with a biology training, this is very obvious, but, for somebody with, let's say, a bioinformatics or informatics training who wants to analyze data, they might not immediately see that, from the paper, this represents the same concept. So Go uses synonyms extensively and description of the concept very clearly in order to make sure that, these things are captured. So it helps to understand differences and similarities between concepts and, scientific phrases that you can find in the literature. So another, the opposite side, one concept can have many names, but one name can represent many objects or concepts. So we have two kinds of eggs, an ovum and a, egg from a chicken here. And so the ontologies, so here we have two different ontologies, the cell ontology, because this is a single cell, defines, an egg cell, and Uberon, which represent anatomical structures, has its own definition of an egg, and so this helps clarify the concepts. Another way the concept is disambiguated is with the hierarchy. So, like I said, the ontology terms are organized in a logical structure. And so, in this case, the egg cell is a female gamete and is a eukaryotic cell and is a cell. whereas the shelled egg is a whole embryonic structure, and in Uberon is defined as gross anatomical part. So both based on the textual definition and on the hierarchical definition, you can see that these concepts are different. And so this is just want to make sure that everybody's familiar with these concepts, like, because ontologies are organized roughly as trees. And so often we use like either tree, the plant, or family tree as analogies when we talk about different relationship of terms to one another. So the root of an ontology is the most general term of the ontology. Leaves are the most specific terms. Branches are groups of terms that share a parent. A parent is a superclass, technically we call it, is the broader concept of a child, and the child is the opposite, right? Is a more specific concept or a subclass of a parent. And siblings are two terms that share a common parent. Okay, so I just wanted to put that out here because we may use these terms without really noticing, and so at least you have a reference what we mean. So there are hundreds of ontology in the life sciences. There's a site called Obo Foundry, where you can browse all the ontologies that meet, kind of fair principles. So here is just showing, well, gene ontology, disease ontology, human phenotype ontology, CHEBI, which is chemical entities of biological interest, and Uberon, the multi-species anatomy ontology. But so there's a lot more, so just so you're aware that there's a lot of ontologies out there. And even within Go, within the definitions, sometimes we define a Go term using an external ontology, like we use KEBI a lot and we use cell ontology a lot. And also when we make annotations, sometimes we refer to external concepts like chemicals and cells and anatomical structures. So the gene ontology itself, it provides a computational model of gene function. Okay, so it's really focused on the function of gene products. So here I use gene as a shorthand for gene product. Okay, the genes in Go, we have very strict definition of function, and the genes themselves, they don't have a functions, right? They're pieces of DNA on the genome. What has a function is the product they encode, either a protein or an RNA. And so this is the scope of Go. Go doesn't represent the activities of small chemicals or the phenotypes or things like that. So it's really the function of gene products. And so it's split into three different levels: molecular activities, signaling and metabolic pathways, and broader, organized biological processes, and cellular organism level systems. And so there's a function, a process, and a component. I'm going to give examples of that in just a minute. So the gene ontology product consists of two parts. One is the ontology itself, which is the framework by which we describe the biological process and how they're linked, and the annotations, which are the associations between genes or gene products and Go terms. And so different databases, they either represent gene objects or gene product objects. Okay, so that's a little bit technical, but just so you're aware of that, keep that in the back of your head. That maybe it becomes necessary to think about that under certain context, but it's really the annotations are an association between a gene product and a Go term. So here's the link to the Gene Ontology website. It's easy to find. And so just to give you the big, origin story of gene ontology. So back in the late 90s, when the first genomes were being sequenced, several groups working on Drosophila, human, mouse, but also bacteria and yeast, they got together and they said, we need a way to represent names of genes or functions of gene in a common framework, because they weren't able to ask questions like, how many genes are present in each of the genome? They can predict by gene models, but how many genes encode specific types of proteins? For example, transcription factors or signaling receptors or protein kinases. So back 25, 30 years ago, the tool that we had was the names of the genes, and these were very inconsistent. They were, you cannot really make a organization of the knowledge based on that. And they were like not consistent with each other. So some organisms named their transporter one system and another organism named it a different system. So we can really do cross-species analysis. And so they came together and they decided to build this tool so that they can compare. And so other questions that this was able to address is, are there the same number or the same proportion of transcription factors in all the genomes or the same number of signaling receptors? And then, for example, which signaling pathways are found, are shared between which organisms? And then what is the phylogenetic range of a certain function or a certain process? So the organization of the gene ontology allows to address all those. And so more specifically, more formally, this is the three aspects of Go. So Go is divided into what we call aspects, or if you will, three root terms. At the root, there's like either a molecular function, which represents the biological activity. For example, an enzymatic activity or an adapter activity or a transcription factor or a transporter. So this is the function. At the molecular level, what does the gene product do? Secondly, there's a component. Where does the gene product act? So in the nucleus, in the cytoskeleton, in the ribosome, mitochondrion, and so on and so forth. And finally, the third aspect or root is biological process. And so this is kind of the biological module or program that is accomplished by a specific set of the molecular functions. So you can imagine you have a number of gene products that act together to perform some biological goal, like glycolysis or transcription or photosynthesis or signaling or development of a specific organ. Okay, so each gene can be annotated to one or many of the different aspects to represent its like full function. So the Go terms is very precisely coded. So there's an accession number, which starts with Go and always has 7 digits. There's a name or label. The ontology here is the aspect that I just described. So here we have an enzymatic activity. There's sometimes some synonyms. Sometimes there's alternate IDs, and there's a definition. And when we can, we base this definition on external resources. So this is RIA, the enzymatic reaction database. And so we have the participants in that reaction. So each term contains at least an accession, a name, a definition, some parents. And yeah, for enzymes, there's chemical participants. So as I said before, the terms are connected to each other via relations. So there's multiple relations in Go. The main ones are is-a. So for example, Exo's biosynthetic process here is a monosaccharide biosynthetic process. Or, a nucleus is a type of organelle. Protein kinase is a type of transferase activity. And then there's also, part of, which is for example, a membrane is part of its organelle. So most of the relations are those two, but there's also many other relations like occurs in, we can say a specific process occurs in the nucleus. We can say that, something is part of, but we can also say that a biological process has some activities. So there's like a larger number of relations that I'm going to describe where you can see those, but the simpler versions of the ontology only have those two. And And so this is an example that shows how function, process, and component are linked. So this is a biosynthesis pathway for ergosterol. And so they're like here, there's 4 enzymes shown in the square boxes. So this activity occurs in the vacuole, and then it provides the substrate for the next activity, which occurs in a different localization. So you can see that we have function, component, and process for all these genes. Okay, and so this is just drawn together in the pathway, but essentially each gene, ideally, if it's well characterized, we should have a function, a process, and a component. This information is complementary. And so I talked a little bit about the different relations. We have ISA and part of. So the fact that there's only these 2 relations, make sure that so you might have heard that the gene ontology is an acyclic graph, meaning a child can never be a parent of its parent. There should not be any cycles. So this is true only for the Go basic version that has ISA and part of relations in it. If you interact with another version, which is Go, so this is what we call the core ontology, it contains a few more, it contains 2 more relations, has part and occurs in. And has part, because it's the reciprocal relation of part of, you can see it can make a graph, because it can make, a cyclic version. So this is just keep in mind, depending on if you're doing analyses with like R and other programs, if it's important that the graph is acyclic, just so you're aware that, some versions of Go are not acyclic. And then we have Go plus, which is what we call fully axiomatized. So that means that there's relations to like cell ontology, Uberon, Kebi, and probably other ontologies that outside that, these are the main ones. But so in the Go basic and Go, there's only Go terms, whereas this has cross references to external ontologies. So currently we have a release that came out last month, and there's 38,000 terms, 24,000 process, 10,000 functions, 4,000 components. And there's over 1 million experimental annotations, which will be described just after. And there's literally millions of annotations by phylogenetic and electronic methods. So yeah, the next speakers will describe all this process. So another important point is that Go is dynamic. So at every release, we have some changes. We either create new terms, remove some terms. We are trying to stay up to date. The ontology was started in 98. So that's like 27, 28 years. We want to remain current with respect to the state of biological knowledge. And therefore, we need to sometimes refactor whole branches of the ontology that were, not correctly representing how we understand biology right now. And so, so we have like kind of 2 ways we refactor the ontology. One is top down, we notice an area that needs just to be updated, or, bottom up. Many people request terms because they're trying to annotate a specific paper, or they notice a concept is missing, or they are confused because 2 concepts look really similar, and then we try to untangle and decide if we should keep a single term or have 2 terms. And so, this is just the summary slide. So an ontology is a logic-based structure for knowledge organization. And, I think it's important to remember that there's 3 aspects of the ontology: function, process, and component.
So now I'm going to talk a bit about Go annotation and what is a standard Go annotation, and also talk about GO-Cam models. So a Go annotation is made of three components always. There is a statement, which is the association of a gene product with a Go term. There is always an evidence, represented by an evidence code that I will explain more into detail, and the reference. So the statement. So the statement links a gene product to a Go term. And I will give you this example of the insulin receptor. So if you can see, the insulin receptor has a function which is a signaling receptor activity. So the association, the annotation one is a molecular function Go term signaling receptor activity linked to this insulin receptor. And it's located in the plasma membrane. So you have insulin receptor linked to plasma membrane. And you can also link the insulin receptor to the insulin receptor signaling pathway, which is a biological process Go term. So for each protein, we have a lot of association and Go term associated to this protein because like many biological process can be made by one protein. Like if you take a kinase, for instance, you can have a lot of different biological process where it is involved in. So each protein gets a lot of different Go annotation. So for the statement, they fall into three different categories. From the start, you can have the manual Go evidence, which is done by the biocurator, and it's taken directly from the literature, scientific literature. So you can have the experimental evidence, the IGA, when it's inferred from a direct assay from a publication. You can have also physical interaction, like a co-IP experiment. You have another evidence type for it, or IMP, like infer from mutant phenotype. In the second part, you can also have like manual prediction. So it's a biocurator, but it's not directly from publication. It's from a different like method. Like for instance, you can infer Go term from sequence similarity. So it's ESS. For instance, if a human protein has a kinase activity and the mouse is very close to this human, you can assign this kinase activity to the mouse protein. You can also have infer by sequence ortholog in the same way. And the IBA, it's infer from a biological aspect of ancestor. It's from phylogenetic trees, and Mark later on will talk about it more into detail. And the third part is the automated rule-based, which are infer from electronic annotation, and there is no human involved in this type of Go annotation. It's fully automated. And so the evidence code, there is an ontology, and there are about like more than 2,000 eco terms. So for human friendly, we say X, IDA, IMP that represent something, but each of these terms has an echo code. Like for instance, you have experimental evidence, you have the echo 000269. So every code has a number associated to it, and it's an ontology. And for Go, we mostly use 11 of these terms that are represented here. So you get these 6 terms, like experiment, direct assay, mutant phenotype, and you have a high throughput version of all these terms, except for the IPI, because it's physical interaction. So there is no high throughput version of physical interaction. So that's the second part. After the statement, you have the evidence code. And since it's an ontology, there is a hierarchy. So it's very convenient if you want to really to select a group of annotation. So if you see on the left part, for instance, you can select the experimental evidence. So you have a number of annotation associated to it. But if you want to go more into detail or you want to look for specific type of annotation, you can go to direct assay evidence or physical interaction. So you can navigate and you can do some very specific searches. The third part of a Go annotation is a reference that cites the source of information, and usually it's a PubMed ID. So each time we curate, as curator, we associate the Go term with the protein, and we add the pre-made ID where the information comes from. So here it's an example of a manual annotation process. So we have, as a biocurator, a specific tool which is called Protein2Go. So it's an internal tool. So if you look at this protein, for instance, you can see from the publication that it's located into the nucleus. So what you do, we take the accession number of this protein that you can see here, and entity ID, and you put the Go ID term that represents nucleus. Like in that case, it's a nucleus with a specific number. We type in the evidence, like if it's IDA, IMP, or IEP, and then we do the reference, the pen-made ID number. So this corresponds to a manual curation, manual standard Go annotation that we do on a daily basis. You also have like automatic method. So it means that there are some mappings that are made from UniProt, or which is the protein database. And in this protein database, you have annotation features. For instance, enzyme commission. For each enzyme, they are represented with a specific EC number. You have a REA that represents reaction. We have a specific keywords or domains. And all this will do some automatic, will generate some automatic Go annotation. You also have like ensemble orthologs, trig after, and interpro and interpro2go, which are domain database that can also generate some Go annotation. So to show you, for instance, if you take this example of the CDK1 protein from UniProt, so as you can see, there is a REA associated to this protein. So this REA shows that it's a kinase. And from this information, from this REA, it will generate an automatic Go annotation that it has a kinase activity. Okay, and several keywords are associated to this specific protein. So you have kinase, and you have also biological processes like cell cycle, cell division, and mitosis. And this term, they can also generate automatical Go annotations. So there is a mapping file. For instance, as I told you, for the EC numbers, so each EC, so you go from EC1, which is you have a general enzymatic activity, and the more you go deep with 4 number, you have more specific reaction, enzymatic activities. So, and each of this number is associated to a specific Go term. For instance, you have the 271122, it's associated with cyclin-dependent protein serine threonine kinase activity. So you can have access to this mapping file that really explain you from which EC, which Go term is associated to this. So now if you look at the corpus of Go annotation, so from the manual Go annotation made by biocurators, you can see that we have roughly a bit more than 1 million annotation. And it's concerned more than 180,000 publications, so where it's extracted from. And when it's sequence-based prediction, like as I told you, by similarity or by orthologs, the biocurators manually review the characteristic of this protein sequence, and that has generated more than 4.5 million annotations. The phylogenetic analysis-based prediction that Mark is going to explain later on, you see that it generates a bit less than 1 million annotation. And the automatic prediction that I described earlier, which is non-reviewed by human, but you can see there are many millions of annotation generating this way. You have like 1 billion in UniProt. So it's the majority of Go annotation is generated by automatic annotation. So if you want to look at the evidence and quality, so these different evidences may provide more or less noise in certain data sets. For instance, if you look at experimental annotation, you have high confidence and high granularity. And phylogenetic, it's high confidence and medium granularity. While sequence similarity, you have medium confidence because you are not sure. Sometimes maybe the active site is lost or It's medium confidence and high granularity. And when it's automatic, you have high confidence with low granularity, because mostly by automatic annotation, you have less specific term. You have more broad terms. So it's important, I think, to understand this different type of annotation, because when you do some work and using Go annotation for your work, maybe you want to look at the experimental versus automatic, because it's not the same type of result that you might end up with, because there are very different techniques used to generating these Go annotations. So who perform the Go annotation? You have mostly expert biocurators from general databases. So you have UniProt, that are a general protein database. You can have also intact or complex portal, which are databases in UK that deal with protein-protein interaction and complexes. You also have specialized databases, Flybase, that deal with Drosophila, Wormbase, it's a C. elegans database, Mouse Genome Informatics, and Reactome. Reactome deal with signaling pathways. And sometimes you have like some specific funded project on a short time frame. You can have, for instance, your Alzheimer or SynGO, which is synaptic function, that have made some Go annotation for very specific tasks. So to give you an idea, there is roughly now about 150 people that are actually annotate using the Go. And in the past, you have more than 200 people. So it's a lot of people are involved in this project and in curating Go and making Go annotations. There is also a Gene Ontology consortium made of a lot of different databases, like mostly the one I described earlier, like UniProt, Reactome, Panther. So all these databases, they are part of the Gene Ontology consortium, and this is consortium like we meet twice a year. We have a conference and we discuss about gene ontology, Go terms, and how we should go, how we should continue, and which direction should we take for the gene ontology in general. One important thing is like what Go is not. So Go is very dynamic, as Pascal said, and it's not an archive of past findings proven to be incorrect or inaccurate, meaning that if we have annotated 10 years ago a protein that is a kinase which has been shown later on that it's not one kinase, then we remove the annotation. So there is a lot of annotation that can be removed when it's proven to be wrong, or also Go terms, as Pascal said, like sometimes they're obsolete because they don't correspond to what we would like to represent. So it's dynamic. That's the key message. And it only describes normal processes. It's not a disease or phenotypes. So that's also very important. Like if a specific, for instance, E3 ligase is acting in a cancer in a publication, we don't annotate these terms, because if it's a cancer, we just annotate the physiological processes. So annotation evolved. So here you can see the progression of annotation from 2018 to 2024. You see it's pretty linear. It's a growth of annotated scientific publication over time. So it's evolving quite linearly. And one important aspect as well, it's like the changes in annotation can be highly significant. So if you look at the term, for instance, annotated to green 1 protein, you can see that between 2002 and 2020, there have been a lot of different changes. Like sometimes the terms, you have 60 terms associated, and then it goes down to 35, and then it goes to 80, and so it's very moving. So that's really important when you use Go to make enrichment analysis, and you really have to mention like which release you are using, because they can be really different results according to the different release you are using at the time you're doing the experiment. So that's really important to see that it's really dynamic. So that was about it for the Go annotation, standard Go annotation. And now I will switch to GoCam, which is a structure framework that links multiple Go annotation together into a computational causal model of a biological system. So in that case, it's activity center unit. And these units are linked together by a causal connection that I will describe more into detail. And it's whole human curated and computational. And there is the source, like the publication that describe this GO-CAM modeling. So I will take the example of NET4, which is an E3 ligase. So the molecular activity is an E3 ligase. And as you can see on this slide, this protein has different substrates. So it can ubiquitinate TNK2, BRAT1, or DASAP2. And it has a lot of different substrates. And what is interesting is like all these substrates, they are how to say, they play a role in the different biological processes. So for instance, if you look at this FR1 signaling, so you have the receptor that is activated, and when it's activated at the plasma membrane, you have NET4 that will ubiquitinate this protein, and it will lead to the endocytosis and the reduced signaling. Okay, so that's one substrate, one signaling. This same protein can also downregulate calcium channels. So same principle at the plasma membrane. You have activated sodium channel, and once it's activated, it gets ubiquitinated to reduce the channel activity. So another substrate, another biological process. Okay. And another third example, it can also inhibit gene transcription in the presence of DNA damage. So if you look here, once you get UV radiation, for instance, and DNA damage, you have association of the NET4 with the RNA polymerase 2. So the NET4, once again, will ubiquitinate the RNA polymerase 2, and it will degrade it to the proteasome. So once again, it's another biological process. And what's interesting, you saw like the two first time it was in the plasma membrane, and in this time it occurs in the nucleus. So if we summarize this NET4 curation and annotation, what you can see, you have a lot of annotation, Go annotation for this protein, but they are not connected and they lack context. So you have like several molecular functions such as ubiquitin protein ligase activity, RNA polymerase binding, or sodium channel inhibitor activity that I showed you earlier. And it's present in several components. So you have cytosol, plasma membrane, and nucleus associated to this protein. And it's part of many biological processes that I've already described. So, and for, as I already mentioned, like for each of these different, so for overall, for this protein, you have like 33 annotation for the molecular function, which is huge, and you have like 52 different biological process annotation. But as you can see, they have different input, but it's not linked. The different input that I showed you earlier together with a biological process. And at the beginning of the 2000, the Go consortium thought that they could make some composed term, like if they could to describe better what's happening. For instance, you have like cellular response to UV plus negative regulation of transcription for AmiGO2 promoter that will lead to the creation of a new Go term, which is negative regulation of transcription from RNA-pol2 in response to UV-induced DNA damage. So the idea was to better, to make more, how to say, more complex Go term to better describe biology. But it found out that this combinatorial was really hard to manage, and the complexity in the ontology leads to errors and inconsistencies. So most of these terms have been obsoleted. That was the question, like someone asked earlier to Pascal. So that's one of the reasons why so many Go terms have been obsoleted, because they tried to make complex terms, but then they thought like it was not manageable. So we went back to the more simple Go terms, and instead try to represent the complexity using GO-CAMs. So what is GO-CAM? So it's another way, let's say, to provide to extend Go annotations. I try to provide context, like cellular component, like in which cellular component, cell, tissue, the activity takes place. The molecular function captures the substrate, and the biological process is in which biological process this particular molecular function occurs, is part of. Okay, and we define like causal connection between the activities. So how it looks like. So the core of a GO-CAM is a molecular activity enabled by an active entity, which is a protein. It has input a target entity. So for instance, a kinase has input the substrate. It occurs in, there is a location represented by a cellular component Go term, and it's part of a biological process. So it's another. So every statement in this Go-CAMs are standard Go annotation. Okay, molecular activity is represented by a molecular function Go term, and so on. But then, since they are linked together, all these Go terms, standard Go annotation, it gives context for every single step. You have more context than a simple Go annotation. And finally, you have like this molecular activity is linked to the next molecular activity by a causal link to the next activity, and it's a relation. So we have like about 10 relations, like positively regulate, directly positively regulate, directly negatively regulate, provide input. So you have a short list of relations that we use to link this molecular activity together. So here is a GO-CAM model view for the regulation of the RNA polymerase by net4. So net4, you see that has ubiquitin transferase, protein transferase activity as input the polymerase, which is part of the ubiquitin-dependent protein catabolytic process, and also part of DNA damage response. And this activity occurs in the nucleus. So it's a really GO-CAM a good way to put some context in the standard Go annotation. And this activity directly negatively regulates polymerase 2, which has the RNA polymerase 2 activity, which is part of transcription by RNA polymerase 2. So it directly negatively regulates because it sends it to the proteasome where it degrades it. So that's a short example of a GO-CAM model. And here is an example of obsolete precomposed terms, as I mentioned earlier. So you had this large complex term to try to represent biology, but it was too complex to manage. So all these terms, like for instance, regulation of planar cell polarity pathway involved in axis elongation, all these terms have been removed along the way since we start to curate with GO-CAMs. So to that effect, like GO-CAM started around 2018, 2019, I think. And so as you can see on this graph, you have a decrease of the number of Go terms, especially representing the biological process, like all the terms that have been obsoleted. So you went from 30,000 to less than 25,000. While the molecular function terms and the cellular component term didn't change that much over time, over these last 7 years. So I showed you earlier the protein to go, how to make standard annotation. Now it's the GO-CAM visual pathway editor, which is the editor for curator to make some GO-CAM. So you have the activity unit box. When you drag it, it opens an activity unit form. So you have the gene product, the molecular function, the evidence, and the reference. Since it's a standard Go annotation, you always have these three statements that should be here, always, like the Go term, the evidence, and the reference. Same for the biological process, evidence, reference, and same for the cellular component, where it's located into the cell, so with the evidence and the reference. And once you made like several a molecular unit like that, then after you can drag from one to the other, and you have the choice like which relation do you want to put in between those two activity units. And as you can see, you can also use chemicals in GO-CAM. So for instance, you can also add chemicals if there is a specific chemical like that activate a protein or inhibit it, you can also represent it to GO-CAMs. So if you're interested, you can look at the GO-CAM browser that I showed you. There is a link here. So it's made by the Go Central. So you have all the different GO-CAMs that have been generated. Like you have a bit almost 1,300 GO-CAM concerning humans, a bit more than 300 for mouse. And always you have the title, the organism, the different biological processes that are included into this GO-CAM, the cellular component, and the genes. So it can be, if you want to have a look, you have access to this page. It's open access. There's another way to be able to retrieve GO-CAM, which is interesting. It's Index. So Index projects provide an open source framework where scientists and organizations can store, share, manipulate, and publish biological network knowledge. So this website is really for biological networks, and there's different. It comes from different resources, and now they've been putting the GO-CAMs into this index resource, which is very convenient. So that's how it looks like, for instance, for GO-CAM. So you have the different gene, and you so, for instance, you get L-cysteine. So that's a chemical. So this GO-CAM includes some chemicals, and you have a legend of the different relations that are available. So in that case, you can see, for instance, that EGT1 as input is histidine, and you have like description of this GO-CAM in index. And one interesting thing, like if you do like some enrichment, so you can put to the iQuery, so you can put a gene list. For instance, you have a gene list that you have generated, and you can put all the protein into your list, and what you will do, it will give you the number of genes from your list that are present in the GO-CAM, and the GO-CAM, it will highlight the protein present in your list that are in the GO-CAM. So for instance, if you put the DES receptor text list in the query, for instance, you will see that it will pop like this GO-CAM, like involvement of Cas7 in apoptosis, and you have 3 genes from your list out of the 6 that are present in the GO-CAM. So you can see if some specific pathways are enriched in your list that you provide to index. So it's very convenient if you are a list, and if you want to look at GO-CAM that are, if they are present, and if they are in your list some specific biological processes that are overrepresented. So to finish this part, so you have a standard Go annotation that I described, which is one term, one gene, while you have a Go-Causal Activity Models that are multiple connected annotation. And one important thing is, like, since Go-CAMs are made of Go annotations, every single statement, every single standard Go annotation that is made for a Go-CAM, you will also have them in the protein page for standard Go annotation. All the annotations are also transferred to standard normal Go annotation. And one important thing is that all statements are supported by evidence, and standard annotation and GO-CAM model, they can be viewed and downloaded in AmiGO, QuickGO, GO-CAM browser, and Index. And now I will discuss more about AmiGO and QuickGO. So if you want to look at some specific Go data, they have several ways to do that. You can look at them in AmiGO, QuickGO, and UniProt. So we start with AmiGO. So it's made by the Go Central Ontology. So it's an annotation browser. So you can search for a specific protein to see all the annotation, or you can search for Go terms. You have a simple search, quick search, and you also have more complex search. You can search through PubMed, you have some advanced search, or you can directly browse the ontology. So let's say you want to look at a specific Go term. So you type in the Go term from this quick search. Like, let's say you type toll-like receptor signaling pathway. You arrive to a specific page of this Go term. As Pascal mentioned, you have the accession number, you have the name, the ontology what it belongs to, if it's a biological process of molecular function. You have synonyms and the definition of this specific Go term. And you have also extra information like the annotation, like where this Go term is present in which protein. So you have the list of the protein where this annotation is present. You have also a tree view that I will describe later on. And you have additional information. So now, let's say like you have typed in the search toll-like receptor signaling pathway. But if you put a space, which is very weird, but when you put a space after this search, you will have another page. You will not go to the definition of the term, but you will have a broader search result. So if you put a space after this specific Go term, you will have like the ontology. So it means that you will find a list of all the different Go terms that belongs to that possess this specific search, but it's not restricted to these specific terms. Okay, you have other Go terms, for instance, and you also have like the gene and gene product associated to this Go term, and then a number of annotations that you can access. So one cool thing about AmiGO, it's like you can search, you can use filters, for instance, like, okay, you have your toll-like receptor annotation, and you find out that you have like 724 different annotation, and you can search for, let's say, you say, okay, I would like to have all the annotation of humans. So then you select organism, and then you go Homo sapiens, and you say, okay, you have 96 annotation for this Go term. So you have 96 proteins that have this specific Go term. So you can search by organism. You also can search by evidence. You say, okay, I would like to have all the annotation, but only experimental evidences taken from paper. I don't want like automatic Go annotation. And then you go and you select like experimental evidence, and then you end up with 85 evidences. And you can also combine the different filters. And you say, okay, I have such annotation. I choose experimental evidence in human, and then so you can play around and really make some specific selection for the annotation you really want to look at. Then, as I told you, we have the infer tree view. So that's the above term. So, for instance, you have pattern recognition receptor signaling pathway. It's a broader term, and the toll-like receptor signaling pathway is part of this pattern recognition receptor signaling pathway. So you get like the broader term from the hierarchy, and you also have the child terms. For instance, you have toll-like receptor, like MID88 dependent toll-like receptor signaling pathway is a child of toll-like receptor signaling pathway. So you have a list of the broader term, the more in the hierarchy, and the child. So that was for AmiGO, and you also have QuickGO that have been developed by the ABI group, which is located in UK. And you have also the same type of result, and you can access the same type of data. But I give you both, so you can choose which one do you prefer. If you want AmiGO or QuickGO, that's up to you. But you have roughly the same type of results. So for QuickGO, you have a search. So if you type in toll-like receptor signaling pathway, you end up with a page where you see that you have the Go accession number. You see it's a process. There is a basket you can use and play around. You get the term, and you get the annotations that are related to this specific term. Once again, like it's another version, but you have if you go look at the Go term, you end up with a page where you have access to the definition, the Go number, which type of Go it is, and you have some comment, and you also can have access to the number of annotations. So if you click on the 44,000, you have access to all annotation. And you have an ancestor chart. So in AmiGO, I showed you have a tree, you have the list of the child and the terms that are above in the hierarchy, but in QuickGO you have the exact tree of all the terms that are in the hierarchy above toll-like receptor signaling pathway. So that's the difference between AmiGO and QuickGO. Okay. So and the child term in QuickGO, you have the table with at least all the terms that are direct descendant of this specific Go term. So one interesting part also in QuickGO, you can have co-occurring terms. So it's show that, for instance, if you have the toll-like receptor signaling pathway that are present in 100% of the annotation, you will have 19.75% of this protein that have also inflammatory response. And you have like 2.9 that also have pattern recognition receptor activity. So you can see that in a protein, like what are the terms that are usually come together. Okay, so it's co-occurring terms. And then you can also look at the annotation. Like you type it, you click on the 44,000 annotation and you end up with all the different annotation. And once again, like in AmiGO, you have different way of selecting exactly the annotation you would like to have. Like by taxon, if you want a specific organism, by gene product, by reference, by aspect, by evidence. You have a lot of different ways to search for specific annotation that you would like to have for your specific purpose. And once again, you also have the different filters that can be combined together, exactly like in AmiGO. And you can also customize which data to show. And one good part is that you can export the list of annotation. Like if you want, you have a functionality that can allow you to export. Like for your specific work, you can export anything you want once you have made the filter that you required. So to finish for this part, you have difference between QuickGO and AmiGO. So QuickGO is based on UniProt, which is protein-centric, while AmiGO is gene-centric. And there may be more than one protein per gene, like isoform or other variants. And QuickGO represents many more organisms than AmiGO. So you usually have more data in QuickGO. So to end up this part, you have two main sites where you can look at annotation and Go terms. So you have AmiGO and QuickGO, and it's up to you which one you want to use for your specific goals. And you can search, browse, download, and analyze.
I hope you can see it. So now I'm going to show you a little bit where you can find all these Go annotations that we spoke so much about in genome and protein databases. I'll start with UniProt, which is the group where we are, the project all of us are working in. The website, the UniProt website, is linked here on the left below. That's what you first see when you come to the website, the area where you can search for your proteins, and then a number of panels which give access to several databases, sub-databases provided by the UniProt consortium. The UniProt knowledge base is the one that we are going to concentrate on today, and that's where you're finding all these gene ontology annotations. Most of you are probably familiar with UniProt to some extent, but a brief summary about a few key numbers. The UniProt knowledge base, UniProt KB, is a public database which provides protein sequences associated with biological information. It's freely available on the web and released every 2 months, roughly. We had a release last week, 2026-02, which publishes around 150 million protein records from about 220,000 different species. These are essentially from reference proteomes, which means the protein complements of fully sequenced genomes. UniProt is maintained by a consortium, which consists of about 100 scientists in 3 institutions, the SIB here in Switzerland, the EBI in the UK, and PIR in the United States. And I've put a link to the latest publication on UniProt. So we have 2 sections, and I'm going to insist on this throughout the presentation. SwissProt is the reviewed and expert curated section, which contains only, I'll say, about 0.4% of the records, and then Tremble, the unreviewed section, which contains more than 99% of the records. It's very important to note that although this is a very small number and you might think it's negligible, this set of expertly curated proteins that you can find in SwissProt is extremely crucial for researchers, and it's also used to improve the huge amount of unreviewed sequences using automatic annotation procedures and machine learning techniques. Throughout the website, you can find the yellow icon here for reviewed entries or the gray icon for unreviewed entries. And we recommend that you always pay special attention to this to identify the reviewed information, where both sequences and annotations are validated, generated, reviewed by expert biocurators. A quick overview of the content of a UniProt record, just a few basic concepts. Each record has a unique accession number. It's very important to retain that one. Always use it when you cite a UniProt record because it's unique and stable. This here is a minimum mnemonic just to help you remember which protein it is about, but it's not necessarily stable. So the accession number is really what you should use. You can browse through any record on the website by navigating through the section titles that are listed on the left. And what defines the protein in the first place is, of course, the sequence itself, which you can find in the sequence section. Each record contains one sequence, but if there are alternative isoforms for the same gene, there can be more than one. And in that case, the protein sequence that is shown by default in the entry is called the canonical protein sequence. Complementing the sequence, we have, of course, biological information, for example, in the function section, where you can find biological information either written in free text or using different controlled vocabularies, like the enzymatic reaction here. And Go is, of course, one of these vocabularies, and this section, the function section, is also where you'll find most Go annotations, as we'll see in a little while. And another way of representing biological information is related to important sites that can be associated with a specific position in the protein sequence, such as active sites, binding sites, or post-translational modifications. This type of annotation is also often provided using controlled vocabulary, like here for the binding sites, we're using KEBI. That's the ontology Pascal mentioned in the beginning. And now that I've shown you this short summary of the content of a UniProt entry, and if you look closely at the top right, this is a reviewed entry with a golden flag here. That shows us that we are in the manually curated section. And in the next slide, I will show you how you can understand where the individual different annotations come from and what their origin is. Every annotation, or at least those that were added in the last 12 to 15 years, let's say, is labeled with what we call an evidence tag. These indicate the source, for example, in the scientific literature, or the method that was used to obtain or to build the annotation or to generate a sentence. In SwissProt, the vast majority of these annotations have been added or reviewed by a scientific curator. And the evidence tags in manually curated statements always have this yellow ribbon, and I invite you to always watch out for that one, because it indicates manual curation. These evidence statements or evidence tags are clickable. So for example, if I click on this bisimilarity tag here, I can see a link to the entry on which from which this annotation was inferred. Or alternatively, if I click on this one for publications, I get the list of publications from which the annotation, from which these sentences come or were abstracted from, with the possibility to open the abstract right here in line without having to navigate away. There are also links to the publisher website and to PubMed, among others. Similarly to SwissProt, we also have evidence tags in Tremble, which show us where the annotations come from. The difference to SwissProt is that the annotations have been imported without supervision, without human supervision, or computed automatically by different automatic annotation and prediction pipelines. I've detailed a few here just so you have the names. We don't have time to go into details, but you will be able to find them on the website if you want to find out more about them, specifically those automatic annotation pipelines. Some sites can also be predicted, usually from a combination of PDB import and prediction tools, and then they will have a tag that is called combined sources. And you've probably noticed that for automatic annotations that the evidence tags are labeled with a gray ribbon, not golden as in SwissProt. Now we'll explore how the Go annotations that Patrick described in detail are integrated into this entry view. We're in this human TLR9 entry, where the function section, you remember, contains some free text paragraphs about the protein function, but it also contains Go annotations from the molecular function and biological process ontology. There are two tabs. First, Go annotations, which gives access to the standard Go annotation, visually summarized by this so-called ribbon, where Go terms are grouped in a way that selects, in a way that some selected high-level terms represent a more or less broad range of functions. There are various versions of such ribbons depending on the taxonomy of the origin of a protein, and the UniProt entry view will automatically select the most suitable ones. For example, here we are in human, so it has selected animals and fungi. If we are in bacteria or in plant, it will suggest another type of ribbon with other terms that have been selected as representatives. And following this graphical display, we have the list of terms that are given with their sources and evidences about the contributing team and the publication, if there is one associated. Again, these tags, or at least some of them, are clickable, and we use the same color scheme, gold for manual, and here's an automatic one, gray for automatic ones. And after this list of Go terms, there is a link to QuickGO as well. There can be small differences due to synchronization or also prioritization. If several teams predict or assign the same term, we will only put it once, while in QuickGO you will find it many times potentially. In addition to the Go annotations, the standard Go annotations, we also integrate the GO-CAMs. These were the more comprehensive ways of capturing a protein's function and its implication in processes, as Patrick presented. If there's more than one GO-CAM, you can find, oh, where's my mouse? You can find a drop-down, which allows you to select the one that you want to look at. This one, the one selected here, represents the signaling pathway that involves our protein, which is still a TLRL9 toll-like receptor in human. The pathway that involves the protein all the way down to the transcription factor IRF5. On the right-hand side, we can see all the molecules involved, their relationships, and related cellular components. Like here, it occurs in the cytoplasm. This activity occurs in the cytoplasm, and everything is clickable. The legend is also included. Now, Patrick also mentioned the view of the index website or database. And a very important aspect of the UniProt knowledge base is that it has cross-references to about 150 specialized external databases, one of them being NDX. And if you like their view, you can access the website directly from the protein entry via a cross-reference that can be found in the enzyme and pathway database section. So you can just click there and see the same view as you could have gotten directly on their website. We are still in the TLR9 entry. And after seeing how the molecular function biological process terms are displayed, we'll now move on to the cellular component ones. They are integrated in the subcellular location section of the entry and can be nicely visualized in an image of the cell via this tab here, GO annotation. So this image, if you move your mouse over a term, then it will be highlighted in the image. And the description of the compartment will come up. And again, we see evidence tags and a link to QuickGO at the end of the list. And now some background on how these images are generated. They're taken from an interface called Swiss BioPix, which is a freely available library of interactive biological images to visualize subcellular location data. You can check it out. You can use it yourself if you like. And it gives access to cell types from all kingdoms of life and specialized cells like muscle, neuronal cells, epithelial cells from all these organisms. And here you can get an idea of what type and how different, of what type of cells are available and how different they are. The image that is chosen in the entry view is the one from which most Go terms are available. So it automatically selects the cell that best represents all the terms. Now I'm briefly coming back to something that Patrick also mentioned. It's that one of the pipelines that brings Go annotations into UniProt is using the different mapping files between Go and available controlled vocabulary like the EC numbers or interpro or reactions. These can generate electronic Go annotations, the IEA, based on vocabulary terms that are already in the entries from these vocabularies. But it can also create annotations, Go annotations, which inherit the experimental evidence from the underlying annotations in UniProt. For example, if you have a RIA reaction in the entry that was annotated from the literature, that was experimental, that will cause the addition of relevant Go terms mapped to this reaction, and these terms will then get the same experimental literature evidence tag. Now we're going to look at a few things on the UniProt website that can be specifically interesting in the context of Go annotations. First, we can search for all proteins that were annotated with a given Go term. We're using the advanced search to do that. So click on advanced to the right of the query box. This dialog will come up and you can click on one of those dark blue surfaces and either select from the list or start typing the field that you want to search in. For example, gene, you start typing gene and then all the fields that contain gene will be suggested, including gene ontology. So we're selecting this one, and then we get to the next step. A drop-down comes up where you can type an identifier, or you can also type the beginning of a term, and it will suggest the different terms that are available and that they are annotated. And you can search by evidence, which is very nice. You can select to search for manual assertions, experimental ones, automatic ones, and quite detailed. So if we end up with a query for the term immune response, where this term has been manually assigned, we will get this query. Oh, actually, I didn't want to do that. Yeah. Now, something else you can do, let's say you wanted to compare Go annotations across several entries in a given UniProt search results. In particular, you can add columns for biological process, molecular function, cellular component. To do that, you can click on Customize Columns. And then this dialog comes up with all the section headers. You can either select from these sections, Gene Ontology, click on the arrow, and then select which ones you want to see. Or if you're finding the list too long, or you can't think of which section your concept is possibly found in, you can start typing the concept you're looking for example, Gene Ontology, and then you get a selection of the matching fields, and you can again select them. You can remove columns by unselecting them, or by clicking on the X's here in the top line, which shows you the columns that you have currently selected to be shown. So you can remove the ones that you don't want to see in the given context. Once you have this result, you can download it in tab separated format, among others. So you have all this information included in your table. Or you can share it. You can click on this share button, which will generate you a link to your results that you can share with colleagues or bookmark, or you can use the URL programmatically. Now we're going to show one more thing that you might need for the gene enrichment, gene ontology enrichment exercise, at least with some services. And this is a tool that that allows you to search UniProt with a list of accession numbers, either all from UniProt or from another database. So you have this list of gene names in the shared document for a later exercise, and you may need to or want to find the corresponding UniProt accession numbers for these gene names. So I've used a list of 40 gene names that can be found in the shared documents, supposed to be involved in schizophrenia. Pasted them in here. Then we have two drop-down menus which allow us to select the type of identifiers between which we want to map from gene name we're selecting, and we're selecting to SwissProt. In the exercise, this exercise is about human proteins, and knowing that SwissProt, the reviewed section, has a protein entry for each human gene, we're selecting SwissProt to avoid extra redundancy, and because we're confident that SwissProt is really complete for human. This is not true, not necessarily true for most other species. And as soon as we have selected gene name here, a warning comes up with an additional box here that allows us to specify the organism to avoid a huge number of irrelevant results. And when we click on map, first of all, we select the organism. There's auto completion provided here as well. So we've selected Homo sapiens, and we got the 40 40 results corresponding to our 40 genes. That's actually quite lucky. It does not always work out so nicely. We could have gotten more than 40. For example, if a gene name is the main gene name in one entry, but a synonym in another one. Or we could have gotten less. For example, if the gene name is not annotated in UniProt KB or not annotated exactly in the same way as the source from which our list was taken. We can now download those results, select from several formats. If you want to have a list of accession numbers, like for this enrichment example, you will select list. And now that we've looked at UniProt in some detail, I will, after all, also show that many other databases show Go annotations as well. I show a few examples, starting with model organism databases. Here is an entry view from ZFin, which is the model organism for zebrafish, where we can again find this summary ribbon as we've seen it in UniProt, and a list of terms with their evidences, very similarly to what UniProt showed as well. Now, another organism, similar display for mouse, where the model organism database is called MGI. These were two examples from model organism databases. There are more, and several of these have joined forces and formed a consortium together with Go to provide an integrated view of their data under the name of Alliance of Genome Resources, AGR. And they also show a gene view, this time a bit differently, not the usual ribbon and list view, but they have automatically generated gene descriptions based on Go in the form of several sentences. And if you look closely, you can recognize the individual Go terms again. They have much more data. AGR allows to compare data from the different organisms that the model organism databases represent. You can select which species you want to look at. You can see all data or only experimental ones, and then you can get a table like this where the different, the same gene in the different organism is shown, and the annotated terms come up in the table. This site only shows the experimental ones. Now, if we look across the Atlantic to the NCBI, which has traditionally been concentrating on their own in-house resources, but which now recently has also started to include Go annotations, for example, in Gene ID, also called Entrée Gene. So there's a section, General Gene Information, which includes Gene Ontology. In PubMed, at the very bottom of an abstract page, you can find a section link out to more resources, and Gene Ontology is among them if terms are available, and they show the list of Go terms, of Go annotations associated with the gene that is represented in this paper. Okay, take home for Go annotations in genome and protein databases is that gene ontology annotations are available across a wide range of databases, and that UniProt in particular has various display options, query options, and it seamlessly integrates both standard Go annotations and GoCam with its own annotations.
Now that Go has no more secrets for you, and I hope you enjoyed this first part, we will try to touch another dimension of Go, the fourth dimension, the time. So I will show you that we are not only annotating sequences, present sequences, but with a project that we call the phylogenetic annotation. We can also annotate ancestors. And you will see that it's really useful for Go. This you all know about this tree. This is probably why we all wanted to study biology at the beginning, is how to understand this. What is life, where it's coming from. And Go is participating to this. We are trying to give functions to every gene. When I say function, it can be a molecular function, but it can also be a process. And even the cellular component, the CC, we have these all three aspects. So when I speak about the function, it can be, the CC is not really a function, but I include this in annotation. It's a Go annotation. So what you know is that, of course, every species cannot be annotated manually, even automatically. It's getting crazy to do this. So you have some species, mostly what we call model organisms, for which we have a lot of studies, a lot of papers, and a lot of annotations. And there are some others for which we have nothing. But the goal is not just to annotate, to have knowledge about few species, 20 species or 100 species. The aim, the ultimate goal is to understand how functions evolved and how life evolved. So what can we do with all those sequences for which we have no data? There is something which is really obvious, is you just look if you have similarities, and this is what most of people do. You just make a pairwise similarity search, like a BLAST. For example, here I show you a yeast protein, a human protein. They are very similar. We have a function for the human protein. So we think, okay, we transfer the function to the yeast protein, and that's it. And we might be able to do this for every species, every organism. But it's not as obvious as it seems, because, as we will see, you cannot transfer whatever you want, and there are some limits. To understand these limits, first we have to see the concept of homology, ontology, and paralogy. So, all the genes having a common ancestor are homologous, but these homologies do not have the same value. As you see here on the left side, this is a little tree. It's a little evolutionary tree for one protein, one gene. And we could say we have one sequence in a bacteria, in a plant, and in human. So those three proteins might have the same function. This might be orthologs. So this is simple. But you know that nature is not simple. So what can happen? It can happen that you have a duplication event somewhere during evolution. And for one bacterial protein, you have two homologs in human, animals, plants, whatever, fungi. So this is the question. Do all these genes have the same function? We have orthologs, and when you have several, so the orthologs are the ones, I would say the ones here, the ones having a common ancestor, having the same function. But when you have duplication events, you create paralogs. Paralogs are also related, but do they have the same function or not? That's the question. Some might not. And in this case, you cannot simply propagate the function from one to the other, from here to here, from here to here. This is just an example of a fiction tree that I created. Again, you have the orthologous case where you have 1, 1, 1. And let's say that this family is a family of transcription factors. So they have transcription factor activity. If it's a transcription factor, it's in the nucleus, but only in eukaryotes. Bacteria don't have nuclei, so you cannot propagate nucleus to the bacteria. This is what we call the taxon constraint. Then they might have different functions. Maybe our transcription regulator regulates brain development. Of course, this you cannot propagate it to plants. Plants don't have brain development. I don't go further to the bacteria. You understand the topic. There are some functions in which they are not involved yet. The same, the plant gene might have a transcription regulator close to the human transcription regulator, it's still in the nucleus, but it's involved in a plant-specific process, photosynthesis, which does not exist, of course, in human and in most bacteria. There are some photosynthetic bacteria. Let's say this is E. coli, which does not make any photosynthesis. But what happens when you have a duplication event somewhere during evolution, duplicating animals and plants? You can think that maybe transcription factor is conserved because it's present in in bacteria and everywhere. But again, nucleus, if it's a transcription factor, nucleus is present also everywhere, not in bacteria, of course. But what happens with some other functions like brain development? In this case, so this is a fictional case, we have two paralogs in human and plants. And what you can see here, you can imagine that this brain development function was lost because you have a second copy, and the second copy evolved to another function, which in our case here is bone development. So here we have paralogs which lost the functions. However, in the plant case, we have the transcription factor, and both are still involved in photosynthesis. So you have paralogs that have kept the function. So you see, it's not obvious just to say we have similarities, and so I can easily just propagate function. So how can we do that? So the first big message of that is duplication events are vectors of evolution. They made the evolution. They are really big parts, not only, but they are big vectors of this evolution because they create several copies of a gene. So you have only always one gene that can be preserved, and you keep the organism fitness. And all the other copies, they can mutate, contributing to the creation of new functions and innovation. And this is the motto of evolution somewhere. So you cannot just propagate by similarity some stuff if you take into account the evolution. To go through this, to analyze this, at the Go, we developed a new project. We call this PanGo for phylogenetic analysis-based Go annotation. What are we doing, in fact? What is the principle? The principle is to use phylogenetic trees. We use phylogenetic trees created by the Panther group. You have here the link. So on one side, we have one input is phylogenetic trees. And then we try to associate to this tree the Go experimental annotation. Those have no more secrets for you. You know what this is. The idea is to create, as I showed you, a kind of evolutionary model of functions, how functions evolved in time since the beginning of life. Once we created this model, which I showed you here, once we create this model, every annotation here is translated into a Go term. So this is a way to annotate unstudied species, but taking into account evolution and not only similarities. This is just a quick example. I will show you quite a lot of examples like that and repeat to be sure that the message is really understood, because it's, as we said, it's obvious at the beginning and it's not so obvious when you have to do it. Here, this is part of a tree, a phylogenetic tree from Panther. It's just the upper part. It's a bigger tree. Down you have a lot of, and in green we have annotations to several species. You have human here. You have the mammals, animals, the frog, fish, insects, fungi, and all these have the same function. So we can say, okay, this function was already present at the common ancestor, which is here. Sorry, do you see my arrow, which is here? So if you move this annotation to this node, then automatically you annotate with the same function all those. So this is the principle. This is the principle when it's simple. This is when you have orthologs. But when you have paralogs, I told you already, it's quite more difficult. So we take an example, and this is the example I will take for this part of the presentation. This is a family that contains genes encoding what is called a superoxide dismutase. Superoxide dismutase is an enzyme that is involved in oxidative stress response. It's very important. And in fact, so this is really simplified because all the bacteria are here. In green, we have the planta, we have the fungi in yellow, and the animals in red. So you see, it's really, really simplified. But already here, what you can see in this family, you have 3 times the red color, twice the yellow color, and up to 4 times the green color. So this means that during evolution, something happened here, and there are several duplication events. And you can, by just analyzing this, you can try to localize these events. So not less than 4 events have been identified here. One within planta, even these two are within the plants. The first event separated animals, fungi, planta from animals, fungi, planta. So you have a first duplications. Then you had another one here within animals. And what is interesting here is that when you look the common ancestor for this function, so all these genes from here to here have the same superoxide dismutase activity. So you would say, okay, we can go to the root. The root of the tree is the origin, it's the first node of the tree. Here, the root is the common ancestor of bacteria, plants, and everything. We call this LUCA, the first common ancestor in life. So we could say, okay, let's just annotate the root of the tree to this term. But then there is a problem because all the terms downstairs will be annotated, downstream will be annotated. And we don't want this annotation for those because here we have another function for this node. So after this duplication here, something happened. Again, there was a loss of this function and the gain of a new function. So you see, how are we dealing with this? So we would like to make an annotation to the root, but if we do it, we have this problem. So we have to block it somewhere here and make another annotation. This is the same tree expanded. So this is the real tree. It's exactly the same. You have here. You are here. So these are you have the 3 human annotations. And in fact, again, in blue, we have the superoxide dismutase activity. So you see there are many, many species, many sequences annotated to this function. So again, the common ancestor here is really at the root. But we have this part, this node, you have the red ones. And we have some additional green ones, very localized. These are for subcellular location. So in some species, this enzyme is secreted. And also we have a cross here. This means that it has been shown that here, at this level of the tree, the function was lost, but there was no gain of function. So you see, you have very, very different cases. So what you first do, you think, okay, let's annotate the red to the root of the red. Then the blue to the root of the blue. But then if you put the blue to the root, you will annotate the red also in blue. So then in the second part, you block the blue annotation at the root of the red function. So you have only the red function. For the green ones, we saw that you have only some little parts of the tree. So you can just annotate some little parts. You can just propagate this function to this node or here. You can choose wherever you want. You can choose. You can annotate any part, any node in the tree. But you are really trying to figure out which one is the right one. And also, you block with a not annotation. You block the blue where it has to be blocked, where you lost the function. And this is the final result, which means all these sequences will be annotated to this red function, so this chaperone function. All the blue ones will be annotated to the blue function, except the ones for which we said, no, we don't want this function for this part of the tree. And you have also some little parts where you have some additional information. This is again exactly the same in more simple, again, with the blue function at the root, the red function for one specific node where it's blocked, and the little green parts for which we have some little nodes in the tree where we can say, okay, we have this specific localization here, and of course the nodes where we blocked it. Okay? And this is important, and this is really a take-home message. I will repeat this a lot of time during my talk, but it's really something. When you're used to make blast searches, pairwise similarities, you think the closest sequence are probably the ones who kept the function. This is totally wrong. When you have this is a, a pairwise search, so blast search, pairwise comparison between the three human genes, protein sequences. We are talking of gene. We annotate genes, but usually the functions are the functions of the proteins. So these are the protein sequences. So when you align protein sequences, you have the three human ones, you have the bacterial one, and what you can see is that the highest. I'm not talking about this because, of course, it's 100% to themselves. But when you look, the high similarities are between human CCS, human chaperone, and human superoxide dismutase. And in fact, these two do not have the same functions. But SOD1 and SOD3 have the same function, and the similarities is lower. So you understand the message. It's really not similarity does not mean same function. This is to show you the tool we are using to make this. So this is something you will probably not see in your life. Yeah, if you want to be a curator, phylogenic curator, you will see it. If not, you won't see it. But just to show how we do this. So these are the tools we have. In fact, the principle is, as I told you, you have the tree. This is just the upper part of the tree. We have again the chaperone here. All the others are downstairs. And the tree is always associated with what we call an annotation matrix. We have three matrices, one for the molecular function, one for the cellular component, and one for the biological process. Here, this is the one I show here. Sorry, is the one for the molecular function. It's the matrix here, I cut it. It's a little bigger. And in every matrix, what we have is the presence of little green squares. Every little green square is an annotation that has been done, which means from here we have a human. I'm sorry, the arrow is moving around. The human gene has at least two molecular functions. You have one here for the rat, which is different, and another different here, Drosophila, this is the fly. So, and so these are the annotations that we can choose to propagate or not to propagate to ancestors. We have also multi-sequence alignment. It's very important because you can imagine that if you are looking for uh, important active sites, if the active site is absent, of course the function is lost. So this is something we have also to check when we propagate. And then we have another window with all the links. Elizabeth showed you many links, many databases. So you have links to UniProt, you have the accession numbers, you have links to specific model databases, you have the names, you have everything. This is really useful also if we need some information, you know, to try to understand how this evolved and yeah, to make this evolutionary model. Because what we are doing, in fact, is an evolutionary model. So we go back to our superoxide dismutase family. In the next slides, I show you only the upper part, which is here in red, which means that you won't see all the part, don't see, because if I show you the whole tree, you would get lost. So again, here is the upper part. You have the chaperone node and you have all the other nodes. These are the nodes that I collapsed, that you cannot see the data, but you have data. So, and for this part, we have again the first column. This is the blue function, is the superoxide dismutase activity function. You see that we have at least mouse and rat. And here you have two fish, twice annotation with two sequence in fish. You have a little square here, so you have two. There was a duplication, so these are paralogs, but they didn't lose the function. They both have the function. We take this from experimental data in papers. So everything is passed on papers. Here you don't see because it's collapsed, but you have many others. So again, what we do, we take this green dot and we just annotate this node here, which is the root of the family. But when we do this, we annotate all the family with this function. But as I said, we don't want annotate the annotation for this. So in another step, we look where is the duplication event. responsible for the subsequent loss. We know that here the function was lost. So we block, we say, no, we don't want the first function to go down here. We block it, the flood, but we can add another function. So the chaperone activity here, it's from the, I don't see it. I think that this is the fly. So the fly is used, the fly annotation is used to annotate all these sequences. And when you put this sequence, you have the right function here. And here you have the other function. So the blue function and the red function. So this is how we do it. So again, why is the multi-sequence alignment important? Here is an example. This is another family, it's a kinase family. And when you align, here you have very important sites necessary for the function. And when you have a sequence in which you lose this, you mutate these sites, of course, you cannot put the function because you lose the function. So what we can do is also, when we have this kind of alignments, we see this has been lost. Here it's for these two. We block it here. So we just put the functions upstream. We have the function kinase, but these two, we are sure they cannot have it. So we just click on this node and we say block the function. We can just block the function. And you have several cases where we block the function. And what can you see again is just the function are always blocked after a duplication event. Here, when you have a square, these are insects, and you have the insects again here and here. So there is a duplication. Here it's more complicated because there were several other duplications. But you know, when you have this square, you have a duplication, an ancestral duplication event, and you have one copy lost. You have the same here, this one, you have twice the same in the same species. One which was shown not to have the function, it's after duplication. This one kept the function, this one lost the function. Here we have rat, we have 3 copies, one kept the function, 2 lost the function. So you see, it's really important also to check at the sequence level if you can have some indications if the function is kept or is lost. And this is another very important slide because we are not propagating all the functions that are annotated. What we want to do in this project is to keep only what we call the core function of the genes. So this is an example of a family. This is a part of a tree. You have the annotation matrix here. It's bigger, but even cut it. We have some matrices with up to 1,000 columns, you know. So imagine for human annotation, it's for a curator, sometimes it's not so easy to create the model and to decide which term is good. So in red, I showed you many interesting terms that have been used as a biological process for all these proteins. When you look closer into these terms, you have a little bit from everything, but they're quite related. Some are downstream related terms, some are quite homologous, some are quite synonymous. And the thing is, we don't want to put to all these genes all these processes because this brings a big background. And when someone then wants to know, what is this protein doing, he has to read 10 different terms, 20 different terms, 100 different terms. And they did that, but what is it really doing? So our job here is to, as we have the global view, so we have the view at the tree level, so at the evolutionary level, and we have the view also at the annotation level. So this helps us to decide, okay, one term is the best term. Here in this case, we choose this one, and this one is propagated. Then, you know, Pascal showed you, you have a hierarchy, you have parent terms, you have child terms. And in fact, the thing is, sometimes when you annotate one term, you have automatic annotation of parent terms, and these appear with these white dots. So if you annotate one, of course, you have automatic annotation to the parent term, but only this one should appear, because this is the most important one. So this is a way to clean out a little bit the background. And the table here shows you have a comparison between the average terms existing for all the families and the average terms that are really used in our process. And what you can see here is really, you see we have between 10 and 20% of terms that are only used. So imagine the big background noise that we remove with this process. So not only are we annotating, propagating functions to organisms for which there will probably never be any studies, but also we are choosing the terms we are propagating. We are not propagating everything. And this is another thing which is important. Here I introduced the fourth dimension, the dimension of time. We are not annotating sequences, nowadays sequences, but we are trying to infer when the function appeared in evolution. And this is a heat map that we made. This concerns only human sequences. I will come back to the human annotation a little bit later. And just to show you when some function appeared. And of course, for example, if you take metabolic process, it's a very general term. We have general terms and less general terms. In fact, metabolic process is life. If you have no metabolic process, you have no cell. So this is totally normal that since the beginning, since Luca, we have this. Because without this, there wouldn't be life. And then we can see that there are some peaks of new function appearing. It doesn't mean that the white doesn't mean it's zero function. It's just less. And the darker they are, the more we have a number over average, of appearances. And you can see that, yeah, you have some peaks appearances when you go from prokaryotes to eukaryotes. When you go from monocellular to multicellular bodies. You can imagine that when you have multicellular bodies, you need new functions. It means you have different organs or you need more communication between the cells, so you have new functions. And all this comes from duplication and then evolution. This one is interesting and it's seen in several studies. It appears when you have the organism with skeleton, bone skeleton appearing, and at this level also there was, at the origin, there were some duplication events and a lot of mutations and appearance of new functions. So this is how to draw evolution, And so from trying to annotate ancestors with Go terms, you can also follow a little bit the evolution. With, of course, the latest immune system processes, which came, latest, of course, because it's not present in Luca. So I try to make here a kind of progression. And it's interesting because this is something we've made for human, because human is the showcase. But as we are annotating evolutionary trees, this is true for every kind of organism that is in our tree. So how can you find the annotations? In fact, I will not go back to all the sites, but all the Go sites that have been presented earlier by Patrick, by Elizabeth, by Pascal. You can just find annotation. Here, for example, we have this copper chaperone, and we have the annotation to superoxide dismutase in Cerevisiae, and it's by IBA. So when you have this evidence code, this evidence tag IBA, which means inferred from biological aspect of ancestor, it means that this has been annotated by similarity, but following an evolutionary model, not just by blast. In UniProt, Elizabeth showed you the ribbons. You have also the tag. So you can find easily our phylogenetic annotations. They have that specific tag. So to conclude this part, here are really the most important take-home messages. The first one, which is for me the most important, is similarity does not always mean same function. So when you have no other thing, you can make a blast and you can expect, okay, we have same function. But as I showed you, I hope I convinced you that this is not totally true. It's better to make phylogenetic annotation, which models the evolution of gene function, gain or loss of function. But we include also taxonomic constraints. As the nucleus does not exist in bacteria, you don't propagate the nucleus into bacteria, but it's like photosynthesis in human or many other terms. So you have to have a knowledge, a general knowledge, because you might make mistake. And when you begin annotation, you make some mistakes because you cannot know where every Go term has to fit in which taxonomy constraints. Brain is, what's the limit between brain and neuron? You have organism having neurons but no brain, you know? And this is really something you have to look for to be sure you annotate the right ancestor and not the wrong one. We are speaking about a highly selective process here, which means that we are cleaning a little bit the background. Background doesn't mean it's wrong. I would say it's less right because what you have is more general terms, you have synonyms, you have downstream effects, you know, what we call also phenotypes that we don't want to cover here because what we want is really the clear function of the gene. If you want to make an evolution of the function, you need to first to define what is the clear function of the tree. And as I told you, we are not annotating individual sequences, we annotate ancestors. And also, do not forget, we have our specific tag IBA. This is important when you make search like enrichment. We will see this a little bit later, but it's really important to know how to find our terms. So now, I showed you a nice project, but it's a manual project, and we can also not, even if we increase the number of species, we cannot annotate all the species from the live tree. Of course, this is obvious. So for the moment, in our trees, we have 143 organisms. We choose the organisms which are the most complete, which have the best sequences and the best protein prediction, gene prediction and protein sequences, because it's better when you want to align the sequences for making trees. And yeah, it's also better for propagating functions. But this does not mean that we forget about all the rest. So we have this other project, which is the tree grafter project. What is this program doing? In fact, it's taking any other sequence from any other species and it's trying to put it in one of our trees. And when it finds its place, it's propagating all the annotations which have been put upstream, so from ancestors. So this allows to increase a lot the annotations, the phylogenetic annotations. But this part is not manual. And so we can always put some caution because it's better to have some manual checking. You have the methods paper here described, and at the moment we have several hundreds of million annotations just with this project. So it's a super PanGo, but with less, I would be, more cautious about the truth of this. But it's a nice way to propagate and to feel the nice tree of life that I showed you at the beginning. Now we go a little bit, we are focusing a little bit on human annotations because you know that the human genome, the human proteome, and what we call now the human functionome is the showcase. So what I show you here, it's specifically about human phylogenetic annotation. Of course, again, we are annotating phylogenetic trees, we are annotating evolution. So we could expect exactly the same for mouse, for plants, for fungi, for bacteria. We have this human showcase because we finished annotations of all the families having at least one human gene and having at least one experimental annotation. And this comes to a question that was asked earlier during the course, is what is annotated and what is not annotated. So we created this specific site for the human functionome. You have the address here, so I really advise you to go to have a look. What you have here is first on the left side of the screen, which are the terms which are the most used. For example, nucleus is the CC which is found in the most human genes. Very close are protein containing complex. So we have many complexes, but this is a very general term. It's not really informative. Okay, so you have this. Then you have on the middle here in dark blue, you have an introduction on the site and you have this enrichment tool. I will come back to this one later on. And down here you have all the list in alphabetical order of all the over 20,000 human genes with name and with the annotations. But this is not so easy when you have to find a gene, so it's easier to take the name and to make a search. So when you search, for example, SOD1, the superoxide dismutase gene, then you open another page where you can scroll through. I cut it in 3 pieces because yeah, it's harder to scroll with the slides. So the first section it shows information about the gene and external links to UniProt, Panther. I really advise you to have a look on the site. So this is the site which shows the trees, the phylogenetic trees, and some other browsers, Alliance Genome. This is the human site for human protein specific to human. You have the NCBI site, and you can have these links. And it shows you a summary of how many annotations have been selected, have been done. So this is the number of IBAs, different IBAs you have for this family. So, and here we have only 7. You see, it's really selective because it's quite a big family. There's several hundreds of sequences in total, and we have only 7 annotations. The 0 corresponds to the number of aspects. We know, the 3 aspects now in Go: molecular function, biological process, and the cellular component. If you have 0, it means we have at least 1 annotation for all the 3. And when you scroll down, you have for each process, for each aspect, you have the different annotations that have been selected. We do not have the relationship like in the GO-CAM to see one function occurs in one specific cellular compartment. But in this case, it's not the case because these functions are together, because the superoxide dismutase bind copper. They're involved in the removal of superoxide radicals, and you can find them in these different compartments. And then downstairs you have, for every annotations I showed you, the detailed origin of the annotation. So for in which species you have a paper, you have an evidence, experimental evidence. Here we have 2 papers for human. We have another paper in Candida albicans, so a fungus, showing the same, and so on. So for every of the 7 annotation selected, we have done the origin, which is really important. You have always to be able to trace back the origin of the term, of the annotation. This shows you for human where this annotation comes from. And you see, it's quite amazing because thanks to phylogenetic annotations, you have some annotations of human genes coming from many different parts. You have here in this part, this blue, this darker blue, the direct annotation. So we have direct evidence that the genes have this function. And you can see that this is quite not unexpected, but yeah, a little bit, that many of the genes have their annotations from paralogs. And I just told you, take care, paralogs can have a function, but they can also lose the function. And in fact, in this case, sometimes when you have the 3 aspects, you keep 1 or 2 aspects and you lose the 3rd one, which means they can have the same function, but in different compartments, So you still propagate the function. And you see, just human experimental data from human is just this blue. Then, of course, no surprise, in gray, mouse and rats are really models to study human function, human gene function. So we have a lot of information coming from those rodents. But we have some from some fish, from worm, from the fly. Fly is really a nice model that has been used for years, for decades, for signaling. All the signals coming from the cell. You have the stimulus on the surface of the cell, and you have all the signaling to the nucleus to express some specific genes to respond to the signal. We have annotations coming from fungi. Pompey is interesting because it's a fungus, but it's quite close to human, especially when you are studying cell division. So a lot of cell division stuff comes from the fungi, and then have plants even from bacteria for basic metabolism, which has not changed during evolution, and many others. So you see, thanks to this, we could fill a lot of gaps in annotation of human function genes. And this is already not up to date anymore. This was after the first pass of the annotating all the families having human genes. And what you can see, we have a big part. So on this scheme, over 82% of human genes now have at least one Go annotation. A big third has already all the three aspects, which is quite good. And you have these blue and purple ones, those have already two of the aspects. And then you have those with only one aspect. And there's still a big part, about 3,000 human genes, for which we have no data, which means we have no relevant. Sometimes we can have some annotations, but they're not, like protein binding terms or very general terms that are not interesting for us because it's not bringing anything for the evolution. So we have still these 3,000 that still need to be filled. So this comes probably from the fact that first, there are some evidence, but they have not been annotated yet. The papers have not been seen by a curator, or just that these genes have never been studied. So if you don't study them, if you have no evidence, we cannot do anything. But these genes are in families. We have still a part of about 1,000 genes which are even not in families. So this might be this means that you have even no homologs, even you have a human gene, you have no mice, you have no other homologs anywhere in other organisms. So this might be what we call pseudogenes or wrong positives. Sometimes you predict genes. You have computers, you have processes to predict genes, and sometimes they predict genes that do not really exist. The ultimate goal, of course, I'm utopic because I explained you the case of the white ones, is to have the free aspect for all the human genes for which we know that they exist. So this is really what we try to get. We will never get this because one day we'll reach a plateau, which means there is a moment that, the genes which are not studied, so we cannot do anything about this. So this was just to show you how far we can go just using the phylogenetic annotation. We increase the coverage, not only of human, of all the species, and we increase the specificity. We are highly specific. We increase the selectivity of the terms. So knowing this, now I will just go to the last big part of our course today, is to show you an example of what we call enrichment analysis. I will show you mostly everything focusing on human, using the human side, because as we're highly selective, we have a lot less results and it's easier to show to it. I really advise you to test different platforms. I will show you. So first of all, what is enrichment? In fact, I think most of you know about this. The aim is just, when you change something to a cell, you have a large amount of genes getting up, another amount of genes getting down. You have a list of genes, but you don't know what is inside this. So the aim of the enrichment is to identify the annotations, the processes, the components, which are enriched in these changing genes. So is there, for example, I put some heat, so I warm up a cell, genes get up. So are there some specific processes which are perturbed, which are touched by this heating? And to do this, we need this kind of enrichment tools just to see if we have something statistic, because we can have it randomly. So this is, exactly to show you. Here we have a mix of genes. We do something, perturbation of process 1. Let's say like a perturbation of the red process. So we expect to enrich, we look the genes touched, and we expect that in the touched genes, we have mostly red ones. Of course, we have also other processes. You know that life is complicated, and we have regulations. Regulations are complicated. Processes might overlapping. So you don't have just one process, but you enrich your list by a process. You enrich your genes. You touch genes by a process. This is used a lot of time here, just a few examples. For example, you can compare tumor tissue to healthy tissue in cancer patients. Say, okay, what is expressed in one tissue, not in the other? You extract the list and you look, what kind of process or function is involved in cancer? You can do it with blood samples and look after exposure to toxic chemical, disease mechanisms. Also, you compare expression of genes in a patient having a disease and the healthy patient. You can use it in drug discovery. You know, look for side effects. So it's a very big use of Go, and Go is really important for that kind of research. What are the gene enrichment analysis tool? So this comes from my favorite AI, ChatGPT. I just asked him, please list me. You have many different tools. I think the best ones are here on the list. I advise you to go yourself, to go, to look, to check, to play with it, and to look which one you prefer. They do not all give the same results, as I will show you. I will just talk about the one about the protein, the protein, the human functionome, sorry. There is another one also of the Gene Ontology resource. It's exactly the same window. You do not get the same results because here you have the whole gene ontology. Here you select the IBAs. So this one is highly selective, and this one is only on human. So let's take an example. We have a paper here where they look for transcriptional regulation of vascular endothelial cells in response to hypoxia by HIF1. So, in fact, here I give you a list. This is a list of about 200 genes that are induced in response to hypoxia. So, we expect that the hypoxia response is, of course, represented, but what are really the processes induced during hypoxia? So you take this whole list, you copy it, and you just paste it here in the box, and then you ask to give you the enrichment, and directly I come here to the result. You get this table. You can adjust some values also for the enrichment. I will not go in details because it's already late, and we can. I really would advise you to go through it. But what is important in this list? It gives you groups, enrichment groups. Where is my here? Enrichment groups. Every time you change from gray to white to white to gray, it's another group, you know, and you have some other all the hierarchies. So all these terms are present, but you know the enriched one, the best to see is the parent term, and you have several groups here, and the most enriched groups are glutamate catabolic process, glutamate biosynthase, mitochondrial automembran. Of course, hypoxia is respiration, mitochondrial, and response to hypoxia comes here. What is important in this table, you have different numbers. You have the numbers of genes in total. So in the over 20,000 genes, you have the number of genes present in only in the 200. So you have a difference between the expected values and the enrichment. You see here we have higher enrichment because, these genes are overrepresented in our sample of 200 genes. So what we need is to have the highest value of fold enrichment and the lowest p-value. The lower the p-value is, the more statistically significant that it's not just the presence of those functions is not just random. So just to show you here, I put you the whole because this is the whole result. This is something which is specific to the Pango when you use the Pango. We are very specific. We are highly stringent, so you don't get a lot. If you use some other sites, some other tools, you might get a lot more results. But they're interesting, and for example, here it's angiogenesis. Putting angiogenesis. So angiogenesis is the formation of the blood vessels. Oh, this is also involved. So this is induced in hypoxia, and you have also what is also, would say, a surprise is, yeah, collagen fibril extracellular matrix organization. So the external part of the cell is reorganized also, and then you have some less important terms. And what is interesting again? Ask ChatGPT or your other favorite AI. You can use another one. And just asking, you pass him the list and say, what is enriched here? And this is the result. And it's quite amazing because the three first are the three first that you found also here. The three first that you found also using the tool. And then you have some more. You have some representative genes, and it gives you even a kind of summary. So you could say that, oh, AI, normal AI is also an enrichment tool. And yes, it can be. The only thing here is you don't have the values, you don't have the statistics, and you don't have the source. So it's good to have an indication. It's really nice, it's interesting. But if you want to make a publication, as a scientist, you want to publish something, you need a strong background, you need a strong evidence. So here you don't have the evidence. You have just, some rapid search. So I would advise, if you want just an idea, you can use this one. If you want something more clear, more precise, and you see when the order is not exactly the same, then use one of the tools. So take home message here for this one. I already just repeating what I told you already, is that first of all, it's important to choose which platform, enrichment platform you want. For example, with Pango, we use only the core function. So rather than the broad spectrum effects such as phenotype. So as a result, you have fewer but more specific categories and your enrichment. But of course, I encourage you to compare Pango results across enrichment platforms to ensure results. First, to see if it's reproducible, if some other platform give the same, and to see the difference also, because you don't have the same results. I will not show you all some other results because it will take some more time. The last conclusion about this one is that, of course, ChatGPT and AI tools behave well, but they do not mention important details that you need, statistical details. So to be really rigorous, scientifically rigorous, but they can give you good information. And so just to finish this long course, I have here a list of resources. So we will not go through all of them, but they are here, they are available. And for example, we have further reading. I really advise you to read these links to papers published describing our different projects. So you should find in the papers almost everything that has been discussed today during this course. The options, Go data extraction options, how to download Go data. So all this has been shown also already. The gene enrichment analysis tools online also. So you see, as I said, I advise you go through all of them, check, play, and look what you really, really need for your research. Programmatic access to Go data. And the last important slide is you can contact us if you have any question. If you have a question about how we do it, or if you have a question about the results, if you do not agree with some annotations, we have two links, one for Go, one for UniProt. I wouldn't advise you to contact directly people. Contact these sites, and then the question will be dispatched to the person who is most susceptible to give you an answer to this. And that's it. And I think we can finish here. I thank you very much for having followed us. I hope you learned something. I hope you enjoyed it. And that now you know a little bit better Go. What is Go and how we can use Go? And what are the different aspects of Go? The aspects, molecular function, BPCC, but also the different aspects in causal relation, in phylogenetic annotation, so in time, in space, we are working with space and with time. Einstein is not so far, and I really hope you enjoy. We are ready to take some questions. I think we have still some time. If we can have questions, suggestions, discussions. I think it's open now, and yes, I give the word to everybody. Thank you again.